Plasmids. Plasmids are extra chromosomal genetic materials present in many bacteria. They are physically separated from the bacterial chromosome and they can replicate independently. They have been used as a vector, as a cloning vector by molecular biologists in recent times. Uh, and they're very fascinating molecule. In this video lecture, we will talk about many aspects of plasmids, starting from their what are plasmids to what is their functions, what are the copy numbers, and how do they replicate, and how we can amplify plasmids within a cell, and how we can get rid of plasmids from a cell to understand their role. So let's begin. So here is it. First of all, let us understand what are plasmids. The plasmids are extra chromosomal, double-stranded, circular DNA molecules found usually in bacterial cells, but not in all bacteria, that have capability to self-replicate and yet coexist with the cell. However, single-stranded and linear DNA plasmids are also there. And some plasmids also occur in eukaryotic system, particularly in yeast. We will see about them. So do all bacteria possess plasmids? The answer is no. Plasmids are not even indispensable for the bacteria that possess them. The bacteria can well survive without the plasmid. So it is an extra facility that the bacterial cell gets by having a plasmid to survive in a particular environment. So plasmid confers the bacteria with a phenotypic character which allows the bacterium to grow in some specialized situations, like if the environment is rich with a certain carbon source which cannot be broken easily, then the enzymes may be borne by the plasmid. If it is growing in an environment where the other bacteria release antibiotics, so to survive it has to have a resistant plasmid. For gene transfer, it has to have plasmids. So let's see what are the functions that a plasmid confers within a bacteria. So there are many functions like resistance to antibiotics is conferred by the R plasmids. Capacity to transfer genes from one cell to the other is conferred by the F plasmids. Capacity to release bacteriocins, which inhibits the growth or which kills the other related bacterium, is the function of Col plasmids. Degradation of hydrocarbons is a function of degradative or metabolic plasmids. Gall formation in case of dicots, which has been used in plant genetic engineering, are the Ti plasmids, function of Ti plasmids, or the Ri plasmids, tumor-inducing plasmids, or root hair-inducing plasmids. There are other plasmids like Sim plasmids, which are responsible for symbiotic relationships. There are plasmids for pathogenesis, there is the virulence plasmids. So we will also talk about the types of plasmids and then we will see their functions, associated functions with them also. So here is the bacterial cell within which we can see there is a bacterial chromosome, which is the essential part of the cell. Any cell cannot survive without the chromosome because it contains the essential genes required for the different functions of the cells. And beside that, we can see there are many supercoiled small molecules. These are plasmids. So within a cell, a plasmid remains just like the chromosome in highly supercoiled fashion like this. So if you isolate this molecule, it looks like it's a negatively supercoiled molecule. Here there is only 3 loops have been shown, but it is actually highly compacted and supercoiled molecule. So if you introduce a single nick in one of the strands, this supercoiled molecule will open like this. This is a nicked circular DNA molecule. So now if you run a gel if you do electrophoresis, agarose electrophoresis rather, then what you will see after isolating or purifying the plasmids, if you run a gel, you can see that there are 2 bands. If your preparation it depends on the quality of preparation. If your preparation is very good, you will see this band prominently, and this will be fade. This band will may exist or may not be found in a good preparation. However, in most of the preparation of plasmids, we can see these two bands. The band which is moving earlier, which is moving ahead, is the supercoiled form. It is called form 1. It is this type of DNA. This DNA actually is moving faster because it is compact and smaller, so it can migrate faster within the gel, the pores of the gel, matrix of the gel. The other band which is behind that, uh, about approximately a centimeter behind that, is nicked circular DNA molecule that is called form 2. And if you linearize that molecule by having a restriction digestion for cloning purpose, or many a times some double-stranded nicks are created during preparation, which is not a very good preparation, then what happens, there is another band in the middle. So this has been run in a separate well, because this molecule is probably a restriction digestion. So you will find the linearized molecule of the same plasmid between the form 1 and form 2. It is positioned between them. So this is how you can locate your plasmid is linearized or not, and you can purify that for cloning purpose. And most of the cloning for cloning purpose, normally supercoiled DNA is preferred. Now let's move to the next slide. So what are episomes? Episomes are also plasmids. Plasmids that can replicate independently in the cytoplasm, as well as they can be integrated within the host chromosome and replicate with it. So this is a plasmid. F plasmid is an example of this episome plasmid or episomal plasmids. These plasmids can integrate within the bacterial chromosome, and they make a strain called HFR strain, high frequency recombinant strains, because these strains are capable of transferring bacterial genes into the recipient cell. This is sex pili. With the help of that, the gene is transferred. Normally, if the plasmid does not integrate within the chromosome, it can be transferred. This cell is called an F plus cell, and it can be transferred to an F minus cell like this via this sex pilus or conjugation tube, which makes a connection between these two cells. But sometimes this plasmid can integrate within the chromosome, and in that case, if it is transferred, it will take the chromosomal genes also along with it when it will be transferred to the recipient cells. So these kind of plasmids, which can remain independently as well as can integrate within the bacterial chromosome or host chromosome, are called episomal plasmids. So now, for cloning purpose, what is done, the plasmids that are used for in cloning, usually the host cells are Rec A negative, so that the recombinant DNA does not integrate within the host chromosome. Otherwise, you will not be able to find the recombinant DNA where it has gone. So for that reason, those plasmids are normally not episomal, and they are the host cell is also made Rec A minus to avoid recombination. So now let's see the copy number of plasmids. How many plasmids are present in a cell? There is a single chromosome per cell that we learned in our last lecture. Exceptions are there with 2 chromosomes, with 3 chromosomes, but not more than that, but they are very rare. Most of the bacteria have a single chromosome, but number of plasmids may be more than one, maybe many, up to thousands. Let's see that. The replicons of plasmids. A plasmid is also a replicon because it contains a single origin of replication and a single terminal site. So it is a single replication unit. So the replicons of plasmids are generally different from those of used to replicate the host's chromosomal DNA, but they still rely on the host's machinery to make additional copies. Why this is being said? Because the replicons, they use the origin of replication, which is under control of certain proteins. Whether this replicon will re-replicate or will remain silent, it depends on the type of replicon. So there are 2 types of plasmids based on the regulation of this replication. They are relaxed plasmids and the stringent plasmids. The replication of relaxed plasmids are loosely regulated, and they can replicate frequently, producing many replicons per cell. Therefore, they are high copy number plasmids like P call E1, PMB1, PUC puck vectors. They are also one type of the PBSSK plus. The vectors that we normally use for cloning purpose are most of them are high copy number plasmid except a few like PSC101. So the stringent plasmids, on the other hand, have tight regulation of the replication, and that's why per cell division they replicate relatively slow, and hence they generate less number of copies or replicons per cell. These are low copy number plasmids, as we told. The PSC101, R6K, 15A, these are the PBR322 also is one type of low copy number or medium copy number plasmids, you can say. So now let us talk about plasmid amplification, how we can increase the copy number. Because for cloning purpose, you need to have more and more plasmids to purify from a cell. So when you are going for purification, the cell number as well as the copy number per cell becomes important to increase the yield of preparation. For that purpose, we need to amplify the plasmid within a cell. The replication and amplification of plasmids is enhanced during partial inhibition of protein synthesis by certain antibiotics like the chloramphenicol. A 5 to 10 fold increase has been found in plasmid copy number, but it does not affect the bacterial copy number. This is why plasmids require the pre-existing proteins for the replication, whereas the bacterial chromosome they require and the cell division also, that is they require fresh proteins to be for the replication, and more amount of proteins are required for chromosomal replication and cell division. So when the cell division or the chromosomal replication is little bit in stress, then the plasmid replication will continue, and this can be done by applying some antibiotics, the protein synthesis inhibitor antibiotics. So now what is plasmid curing? Plasmid curing is basically getting rid of plasmid from a cell to understand whether a characteristic is borne by the is plasmid borne or chromosome borne. If you want to know that, you have to remove the plasmid from the cell. This process is called plasmid curing, removal of plasmid from its host bacterium. Various agents that can intercalate between the bases of the DNA, particularly the acridine types of molecules like the proflavin, acridine orange, ethidium bromide. These kind of molecules which have a plateaued structure like this, they can intercalate within the base pairs of DNA, and they can inhibit replication. They can inhibit replication of many plasmids, but without inhibiting the replication of the chromosome. So what happens, why this happens is not clear yet, that why these molecules inhibit the replication of plasmids but they do not affect the chromosome. This is not clear, but because of this, what happens in the next round of cell division, the chromosome replicates and it moves into the two daughter cells, whereas the plasmid cannot replicate, and that's why in one of these cells it is lacking. The other cell gets it from the parent because a single molecule already exists. So when this cell will again divide, one line of cell will definitely be lac positive, but the other lines, the number of lac negative cells will continuously increase. So in this way you will get many lac negative. Here we are talking about a lac as a lac positive as a character, so other characters may be also there. If you want to see them whether that character is borne by the plasmid, this plasmid curing technique can be applied. So now let us talk about the types of plasmids that they exist. So here is a brief idea in this table that how many types of plasmids are there. The first one is the conjugated plasmid, or you can say F plasmids. These are basically the fertility factors, that the F factors, which are responsible for transfer of genes from one cell to the other through conjugation. That's why the name is conjugation plasmid. These are very big plasmids, 95 to 100 kb. In fact, 200 kb F factors have been talked about. The copy number is relatively low because they are very large plasmids and their hosts are E. coli, Salmonella, Citrobacter, etc. And they actually they're capable of they are self-transmissible plasmids, so they are capable of making their own sex pillars, as well as they can transfer their DNA by themselves without the help of other plasmids. So they are self-replicate, self-transmissible plasmids. The next is the R plasmid, for example, the RP4 or the PSH6. We will see the structure of them, all of them, F plasmid and R plasmid and others, in our next slide. So these R plasmids are basically, the name is for resistance plasmids, because they provide resistance against antibiotics of different types. This RP4 is a little bit medium copy number plasmid, is a low copy number plasmid. The size is 54 kilobase pairs. The PSH6 is about 21 kilobase pairs. It is quite large plasmids, and their copy number is comparatively less, and they are found in Pseudomonas and many other gram-negative bacteria. The first one that the R plasmid had been found in the Shigella dysentery from Japan, and later on it was seen that it acquired many antibiotic resistance property, and many bacteria actually have this. Most of them are pathogenic, and it is difficult to, that's why it becomes difficult to treat pathogenic bacteria many a times by a single antibiotic. So they also are capable of self-transmission. They can form sex pillars in conjugation as well, and they provide resistance against different antibiotics like the ampicillin, kanamycin, neomycin, tetracycline, and this is probably gentamicin, tetracycline, kanamycin, this one, PSH6. So different R plasmids will have a different capacity for antibiotic resistance. The next one is the col plasmid. For example, the P-col E1 or CLO-DF13. They have a relatively less size, 9 and 10 kilobases, but the copy number is of medium range. They're found in Escherichia coli, and they are cholicinogenic plasmid. They release cholicins. They produce cholicins or cloacins, which are capable of inhibiting the or killing other related bacteria. So these col plasmids, particularly the col E1, has been used in genetic engineering to design other vectors like the PBR322 or PAC18, PAC19 or PGEM or any different the PET vectors. The basic backbone of those vectors, plasmid vectors, is the P col E1. So now the virulence plasmids. There are some plasmids like the end plasmid or the TI plasmid. The end plasmid in case of humans, and in case of TI plasmid, we are talking about bacteria. They are pathogenic in bacteria. They are not exactly pathogenic. They form tumors in certain dicots, which you can call a disease because it's a kind of deformation, of course. So you can say that it is a hypertrophy kind of deformation. So it is pathogenic in other way. But these bacteria have been exploited for our purpose as a vector because they have a certain DNA element, the tDNA element that we will talk, that is capable of transferring to genes from a bacterium, from one plant. You isolate the gene, you clone there, and that can be transferred to the other plant. So this TI plasmid, tumor-inducing plasmid, they reside in Agrobacterium tumefaciens, a soil bacterium, which can infect dicot plants, and they cause tumor formation, as we said. The end plasmid are 83 kilo base pair plasmid and present in E. coli. They cause enterotoxin production. There are many pathogenic bacteria that possess these virulence plasmids. Metabolic plasmids. They are very significant. They become significant when Professor Anandamohan Chakraborty, when he designed a bacterium called Chakraborty's bug way back in 1980s, and that bacterium was capable of eating so many different kinds of carbons, about 7 different kinds of carbons and phenolic compounds that bacterium was capable of utilizing and decomposing. So that was a type of metabolic bacterium or degradative bacterium made particularly to clean environment where oil spillage or different kind of phenolics are released in the environment. So there are CAM, PCAM or PTOL. Different names can be given because this can break camphor, and this one can break toluene, And the molecular weight is very high. For the pCAM, it is 230 kilobase. It is not known about the copy number. Pseudomonas and Pseudomonas putida are known to possess them. This can break camphor and this can break toluene, as has been given here. So these are the different types of plasmids. There are others. Sim plasmids are also there. Ri plasmids are there, and there may be many more plasmids. But a list of some of them have been enlisted here. So now let's see the structure of these plasmids. Some of them. This is R plasmid. The R plasmid is the resistance character bearing plasmid. They have a size of about 106.7 kilobases, and they have two segments. There is a big segment called RTF, resistance transfer factor, and there is another fragment that is called R determinant. This R determinant contains the antibiotic resistance genes of different types. There may be multiple antibiotic resistance genes present, and this has been obtained through horizontal gene transfer through conjugation. The first bacterium that was, as I told in a little earlier, that the first bacterium was Shigella dysentery, which became antibiotic resistance in 1950s in Japan, and the bacterium was found to possess the plasmid R plasmid. Later on, it was seen that this plasmid is present in many pathogenic bacteria, which have become antibiotic resistance. So this is about R plasmids. Now let's talk about the F plasmid, the fertility factor. The F plasmid is a very big plasmid, about 100 kilobase pairs, 94.5 kilobases has been shown here. It has a gene 25 kilobase pair region called TRA region, which is responsible for its transfer into the recipient cell. There are many transposons, simple transposons like IS3, IS element insertion sequences, IS3, IS2, and there is a TN element also, TN transposon, TN1000 in the bacterium. They have different functions, and this plasmid is responsible for transfer of bacterial genes from one cell to the other. So they are basically responsible for recombination in bacterial system through conjugation. This is P called E1, the bacterium, the plasmid that is being used as a backbone for many different plasmid-based vectors like PUC8, PUC9, PUC18, PGM, etc. So this bacterium contains genes for colicins. They have incompatibility factors genes also. So now this one is a TI plasmid, which is quite big plasmid, 200 kilobase per in size, and this plasmid has been used. We are talking about the tDNA element. This is the tDNA element surrounded by the left border and right border. This tDNA element contains many genes for auxin synthesis as well as cytokinin synthesis. These are the hormones. That's why if this DNA element is transferred to the plant cell and it gets integrated into the plant genome, what it does, it synthesizes enzymes responsible for IAA and cytokinin, IPTZ, IPT, isopropyl thiogalactoside synthesis, responsible for the synthesis of cytokinin. So in the cell, concentration of IAA, auxin, and cytokinin increases, which are growth hormones, and because of that the cell starts dividing and it forms a tumor-like growth over there. So what scientists have done to use this as a plasma, as a vector, you can replace this tDNA with any other desired gene that will also be transferred, and you can, from outside, you can add auxin and cytokinin according to a desired ratio to generate callus or to make your callus rooty or shooty. This is the vir region because of which the plasmid shows the virulence character. There are many vir genes, about 7 to 8 vir genes are there, which actually make connection between the plant cell and the bacterium. The sense that the injured portion of a plant cell where the bacterium will deliver the DNA. So all these functions, the transfer of DNA to the host bacteria, host plant cell, is governed by this vir region of the plasmid. So this is about the TI plasmid. They have opine genes, which when enter into the cell, they synthesize certain amino acids which the bacterium feeds on to increase its number. So this is about TI plasmid. So now let's talk about plasmid incompatibility or compatibility. What is this? Two plasmids of related character cannot exist in a cell. This is the thing, this is incompatibility. Pairs of closely related plasmids cannot be maintained in a single bacterial cell at a time. Such plasmids are said to fall into one incompatibility group. So there are many incompatibility groups. They have been classified according to that, and so that people can understand that these two plasmids can be incorporated in a single cell or not. So they have been classified. The incompatibility group has been made. So plasmids with different incompatibility group, however, can coexist. For example, the F and called E1. These two plasmids, they belong to different incompatibility groups. So they can be maintained together and replicate independently in a bacterial cell. The most frequent reason for two plasmids being incompatible is that they both possess a similar kind of replicon with same specificity for rep protein, replication related protein, or controlling elements to bind. That's why one of them will be excluded. However, incompatibility can also be due to other types of competition, for example, between the same or closely related partitioning systems. So how the plasmids will get partitioned during cell division. If that is different, if that is common, then it is a problem. If that is different, then both of them can coexist. Incompatibility may be reciprocal. When you forcefully insert two plasmids of same incompatibility group in a cell, then what will happen? A competition will arise between them. And which plasmid will be eliminated? It's a reciprocal process. Any of the plasmids can get replicated, they get eliminated during cell division. They have equal chance, same chance of being lost. Or it can be unidirectional also. If one of the plasmid has certain advantage, additional features that give it an advantageous position over the other, for example, possession of a second replicon, then that plasmid will replicate in number, will outnumber the other and will survive. The other will get eliminated. So this is how the plasmid incompatibility group works. Plasmid, promiscuous plasmids. So before that, let's learn plasmids play important role in gene transfer through conjugation. On the basis of this transmissibility, there are three types of plasmids. One, that is conjugative plasmids. They can make physical contact between the two cells, but they cannot transfer the DNA. Mobilizable plasmids, they are capable of transferring the DNA, but they are not capable of making effective contacts. Self-transmissible plasmids are capable of both. They can make effective contacts as well as they can mobilize the DNA also. For example, the F plasmid. So, a plasmid, if a plasmid is capable of doing both of this, that plasmid will successfully be transferred to the other cell and can transmit certain genes also with that. So that plasmid is capable of carrying a mobilizable plasmid or a conjugative plasmid, which is not capable of doing these two functions together. So that this plasmid can assist in the transfer of the other plasmids, which are partially efficient. Plasmids are transferred between relatively close relative species of host bacterium. That is also true that one plasmid will be transferred to the other bacterium. That bacterium will be either of the same species or strain or will be very related bacterium. This is what commonly happens, but certain plasmids are capable of transferring their own as well as can co-transfer other plasmids with them to a distantly related host cell, host bacterium. Such plasmids are called promiscuous plasmids. For example, the R388, RP4, PKM101 are the examples of promiscuous plasmids. RP4, which falls in the incompatibility P group, can transfer themselves and mobilize other plasmids from E. coli to any gram-negative bacterium. So what function they do, we will see that the promiscuous plasmids carry their own. How do they do that? Normally what happens when a plasmid, or any other self-transmissible plasmid that is transferred, that is not promiscuous, when that plasmid is transferred to the recipient cell, it is transferred in a single-stranded transfers a single-stranded molecule which gets replicated over there, becomes double-stranded using the machinery of the recipient cell. The primase, the DNA polymerase, whatever it uses, all of them is of the recipient cell. But these promiscuous plasmids, they carry their own primase genes when they are transferred to the other cell. That is the interesting feature. And hence, they do not depend on the host machinery, at least for priming process. When the priming has been initiated, the replication, other part of the process can be done by using the cellular machinery. So they transfer the primase along with the single-stranded DNA that replicates in the recipient cell after entering to the recipient cell. Other plasmids actually rely on the cellular machinery. These promiscuous plasmids probably play important role in bacterial evolution. How? Such transfer process, because they transfer genes from a very unrelated bacterium to an unrelated one. So that's why character change or maybe in certain cases we find similarities because of this reason. Because of that, genes with related functions, similar character, exist in phylogenetically distantly related organisms because of this promiscuous plasmid. Okay, this because of this promiscuity present in a plasmid. These R plasmids are example, one type of promiscuous plasmid, and probably because of this nature, they have acquired many different antibiotic resistant genes from bacterium like actinomycetes. So this is because the R plasmid, their multiple resistant genes are obtained through promiscuousness of the plasmid. So how do the plasmid replicate? Plasmid mostly they rely heavily on the host for the replication. DNA pol 3 is required for replication of some plasmid, but some other plasmids use DNA pol 1 for the replication. For example, if you see pol A mutants of E. coli, a plasmid can replicate within that normally, but col E1 fails to replicate. Why? Because pol A is a mutant of DNA pol 1. DNA pol 1 becomes non-functional. So that means the F plasmid requires DNA pol 3, which is normal in this pol A cell, whereas the col E1 fails to do so because the DNA pol 1 is defective. All plasmids examined so far replicate semi-conservatively, like the double-stranded DNA, chromosomal DNA molecule, and remain circular during replication. However, there are significant differences in pattern of replication. Some plasmids replicate unidirectionally, while others bidirectionally. Interestingly, the plasmid RK6 replicates first in one direction and then later in opposite direction from the same origin of replication. So there are variations among within the plasmids about the pattern of replication. Replication occurs by so-called butterfly or rabbit's ear model. This type of structure is formed in which the replicated region forms a butterfly rabbit ear-like structure or butterfly-like structure, and the non-replicated region forms a supercoil. It remains in a supercoil structure, which later, when the replication fork proceeds, it gradually takes the shape of a theta model. So the theta model is this is the way the plasmid actually replicates, and this is an intermediate structure that is formed, which is called rabbit's ear model or butterfly model. So this is the last slide. We will talk about the eukaryotic plasmids. It is not that the only bacteria possess plasmid. Plasmids are not unique to bacterial system. They're also found in eukaryotic system like in yeasts and chlamydomonas. So what type of plasmids are found in yeasts? There are two types of plasmid. One type of plasmid is its indigenous, which is naturally present in the cell, and the other is artificial, that has been made for genetic engineering purpose. There are artificial chimeric plasmids. So naturally occurring plasmids, among them the 2-micron circle, the 2-micron plasmid found in the Saccharomyces cerevisiae are very important, and they are called 2-micron because their contour size is about 2 micron. And they have also been found in some other species of Saccharomyces, like Saccharomyces italica, Saccharomyces diastricus, and Torulactis datila. They also possess these 2-micron circles within them, and they have been studied a lot. In this video lecture, it is the capacity of this lecture will not include the details of these plasmids so far, but a little bit of them, since it has already been a long video. We will talk about this in a separate video lecture in details. There is a 3 micron plasmid also found in the Sisal Saccharomyces pombe. There are certain linear plasmids exist that in the PGKL1, PGKL2 in Cloiveromyces lactis, and PPAC L1 in Prischia acacia. So these are the naturally occurring plasmids in yeasts. So this is a structure of the 2 mu circle which has been talked more, and they contain genes, different genes, rep1, rep2, origin of replication, D element, flip element. We will talk about these in details in separate video. So artificial chimeric plasmids may fall into 2 categories. One is the integrating plasmids, those which integrate within the host chromosome, and the others that can replicate autonomously, autonomous replicating sequence possessing plasmids or episomal plasmids. They do not integrate within the cell because they contain the ARS sequence extra. In addition to the integrating plasmids, they contain ARS sequence in extra. That's why they can be maintained independently. The integrating plasmids include the YIP that contain the PBR322 backbone as well as the his3 site. So they will integrate in the his3 site of the chromosome. PBR322 has been cloned. The backbone of that has been introduced in this vector to make a vector in which you can introduce certain gene also. PYE Liu10. This contains a P called E1 backbone, and it contains the Liu2 site, so it will be integrated in the Liu2 site of the host chromosome. So this is the structure of YIP5, which is one type of this type of integrating plasmid. So now let us talk about the ARS plasmid or episomal plasmid. They are of 3 types, in fact. In total, if you group them, you can categorize them in 3 types, depending on the ARS that is being used. Rest of the part is same as that of integrating plasmid, but the ARS, you can obtain the ARS from the chromosomal DNA, from the mu circle, or from the mitochondrial DNA. Depending on that, which ARS is used, there are 3 types of them. This episomal plasmid. When 2 mu circle is the ARS of 2 mu circle is used, it is called YEP. The backbone is PBR322 in all of the cases. When the chromosomal ARS is used, it's called YRP plasmid. When mitochondrial ARS is used, it is called YIP again. So there will be different numbers behind them depending on their types. So this plasmid is using 2 mu circle as an ARS. This ORI is the ARS of 2 mu circle, and this is the PBR32 backbone, and this is the rest of the part of the chromosome, the LEU2 containing gene. So this plasmid can be maintained independently because of the ARS present, and it can shuttle within E. coli as well as in yeast because it contains the PBR322, and also there will be a replication, origin of replication within it. So this one is the chromosomal ARS containing episomal plasmids of yeast. Um, so now there can be another kind of episomal plasmid that contains the centromeric region. The centromere, and in addition to that, the ARS, which is adjacent to the centromere, that has been also been cloned to make a episomal plasmid. For example, this YCP50. So there are many such eukaryotic plasmids present in Chlamydomonas also. We will talk about these eukaryotic plasmids in a separate vector here, a separate video. Here I have tried to to compile a video for entire plasmids, their types, their functions. I believe that you must have got a good idea about plasmids as a whole. Um, Please do share this video lecture, subscribe it, and press the like button as well as the bell icon to get more such videos. And thank you so much.